Welcome to Revolution Against Evolution. I'm your host, Doug Sharp.
Co-host Rich Gear here as well. And Doug, we're doing a lesson in biochemistry, which means, although you don't generally do it, I'm the big mouth in this group. You're going to do most of the talking today.
Well, I'll see what I can do.
You know that doesn't always work out that way, but we'll do the best we can because he's the biochemist. He's the one that really knows this stuff. So anyway, you want to start out with Linus Pauling? Is that what you wanted to do, or you want to go somewhere else?
Well, I would, Linus Pauling, I think, is one of my heroes, and this is a picture of him. He's kind of a interesting character. He's the vitamin C guy, if you remember that. But he wrote a book called Nature of the Chemical Bond, and so I was able to actually understand a lot about chemistry when I was in high school, and so Linus Pauling, he was kind of my hero. And I also got one of my heroes right here with me, is Derek Marshall. He's written a book called The Chemistry of God. And I want to plug it because he's gotten some things out of the word of the scriptures. He's studying a study of mathematics found in the Bible. And I find his approach very fascinating. I can't claim I understand it all. I'm continuing to unpack everything that he's saying. But this is a good book if you want to digest it. It's available on Amazon, and I hope you look it up and enjoy it. But anyway, chemistry has been one of my passions. Ever since I got a book from my uncle, is called The Chemistry Made Easy. And so Chemistry Made Easy, I learned a lot about chemistry, and I like little tidbits like zinc is the only metal that forms a white sulfide. Why would you ever need that or want that? But that's something that you learn in chemistry.
And I'm not sure I even know what that means, but that's okay. Anyway.
But one of the things I was learning about Linus Pauling, he actually took the way chemical bonding works and unpacked it for me, so I was able to understand it quite a bit. There's actually several different kinds of bonds that are in chemistry. And the first one I wanted to talk about is covalent bonds, and I'm going to do that first. And then I'm going to come back to these other weak bonds that I just showed you here.
Is covalent bond then a strong bond? You said something about weak bonds.
Yeah, this is more of a strong bond. And so let's start with the carbon atom itself. Carbon is a very unique element, and it has 4 different ways that you can connect other atoms to it, and it does it by sharing electrons. And so the orbits of the electrons sort of pass around both elements, and that forms the bond. But there was a fellow by the name of Schrodinger, and he has his famous Schrodinger equations, which I studied in physical chemistry, and those formulas are very complicated. It's based upon the fact that if something is moving at a very microscopic level, it's actually got a waveform associated with it. And so if you pitch a ball at 100 miles an hour, you can actually calculate the waveform that it makes as it travels towards the batter.
Is that the one that goes like this or something?
Yeah, the waveform goes like this, except it's so microscopic, it's at the angstrom level that you can't see it. But it's a principle that applies itself in chemistry, because at the molecular level, that causes certain electrons, as they move around the atom, to behave in a certain way. Now there's a orbital at the beginning, it's called the s orbital, and it's where you have 2 electrons orbiting around the nucleus of the atom.
Okay.
And it, since the waveform is actually in sync with the circular shape, it forms a circle. I mean, the sort of a sphere around the atom.
Oh, I see.
But the second level, quantum level, is where you have what's called the p orbital, and the carbon atom has these 4 places where it bonds, and the p orbital looks like this. It's sort of like a figure 8, and it goes on the x, y, and z axis. And so when you if you connect two of the atoms together, 2 carbon atoms together, and you form this where they connect together like that.
Okay, that looks more complicated than the double bond. I don't get that.
Okay, the double bond, well, what you have is that you're eliminating some of these little orbitals here. Now,
Okay.
you've got these orbitals here that are still available to connect, and usually there's a hydrogen atom sitting on the ends of these when you have a molecule like that. But here you have a double bond, and you have just these left, so that you have this double bond here between the 2 of them, and then a triple bond looks like this. All right, and the triple bond is where you have all 3 of those orbitals connecting right in the middle, and then you have some of it coming out the other end here. So it's kind of an interesting way that things work, and it's basically the atoms are connecting because they're orbiting around each other. So if you have a carbon and a hydrogen that's in a covalent bond, the orbits are shared by the 2 atoms. And I think I've lost Rich.
Yeah, I'm kind of going, so why is that important?
Why is that important?
Yeah,
Okay.
how does that relate to creation? How does that relate to God? I mean, except for the fact that it's organized, and you can see the design in this, but I don't quite understand it, because obviously chemistry is not my thing, you know?
Yeah, eventually what happens is that then you see that you can make all sorts of things with carbon. And
so those 3 were all carbon things.
Those are just carbon things, how the carbon bonds work, you know.
Okay.
You have a single bond, a double bond, and a triple bond. And generally you see single bonds in any of these molecules I'm going to show you.
Okay.
Now, we also have other things that cause bonds. These are the weak bonds. You have, and if you're making a protein, proteins fold up into a whole configuration, and you have things like disulfide bridges, hydrogen bonds, hydrophobic bonds, hydrophilic bonds, ionic bonds, and van der Waals attractions. And those are weak bonds that also affect the shape of a protein. And what you have when you have a protein is that they have there's 20 amino acids that make up proteins.
So Doug, we're talking about things that are involved in life forms.
Life forms, yeah.
Okay, because carbon is in rocks, right? I mean, you got, right? I mean, carbon is in things other that are not living. But right now you're talking about things that are that help you organize, because protein is living stuff, right? I mean,
proteins are what you find in life.
Okay, good. All right. I'm not totally lost then. Just only partially.
Yeah,
Keep going, all right? I'm trying to make this interesting for those people like me who are really in the art forms, not the chemistry and science aspect of it, okay?
Well, I'll go back to my high school days and college days when I was given a project to do, and my project that I chose to do in this Honors Institute class I took at Lansing Community College while I was still in high school.
Right.
I decided I was going to take the origin of life experiments that Miller-Urey had, where he had this little mechanism going on where he had a flask that had water in it and boiling the water, and it went up this pipe here into this chamber where he had other hydrogen, methane, and ammonia, and in this chamber he had a spark. And the spark caused it to create some stuff that got collected down here in the bottom here in this little chamber, and then they siphoned it off and they analyzed what they got.
Okay.
Well, there were some amino acids in there, and they said, "Aha! We have found how life
was
formed."
created. Yep. All right.
And so that's why you send out probes to
Beyond
Europa
Pluto.
and out in the outer space to find methane, ammonia, and hydrogen gas.
Right.
Because they theorized, well, that was the start of life.
Well, didn't they propose it? It was our planet must have had a methane atmosphere because oxygen is a destroyer of anything really being created. I mean,
Right, yeah.
as oxygen, this whole idea of free radicals and rust, and it's all got oxygen. It breaks down everything.
Right.
We know that without oxygen, we're dead. So that's the kind of the oxymoron that somehow it went from a methane atmosphere to an oxygen atmosphere. Of course, we know empirically the earliest rocks, based on their scenarios, are oxygen impregnated.
Yeah,
so that doesn't make any sense.
They call it the Great Oxygenation Event.
Of event, yeah, and it totally.
And they don't
bogus.
actually have any explanation
There's-
of when
yeah.
how that happened.
Yeah.
But it is necessary for them to.
Or any evidence that it did happen. That's what I'm getting at. Yeah. So
But it's
all right.
necessary for their theory to work. So, in order for the theory of abiogenesis, which is the origin of life, and they've most of the people who are involved in this have discovered some of the same things I did, is that it really doesn't work. And so they say, well, that's outside of the realm of evolution. We don't even deal with that.
That's always the
anymore.
workaround. When you actually catch them and it doesn't work, they go, well, that's not really evolution. Evolution is this, this, and this. And I go, yeah, but okay, if I didn't catch you, you'd keep saying it starts here. You basically have abiogenesis. We know that everything we see, life always comes from life. So there has to be what's called the uncaused first cause.
So this is my experiment, okay? I
All right,
said,
let's go back to that.
what we need to do is to see what happens if we add some more zonking to the mixture, okay?
Yep.
And so my idea was to take the amino acids that we created now, put them back in the mix here, then we're going to zonk them some more and see what happens to it, and we're going to get some...
Zonking is more sophisticated than
We're going to
zapping.
find them,
Let's not
find
tell you
those
that.
peptide bonds connecting together, and you have the string together to form these proteins. Well, one of the first things I found out was that if I have alanine and glycine, which is the two of them, the two simplest ones, and I'll show you one of them here. This is glycine, and it's just this little. And I'll show you a little bit of the configuration of how amino acids work here. So
Now, amino acids based up on these bonding things. Is that how that works? I guess how do the bonding that we just talked about related to these amino acids? How does that work?
well, those bonds I'll show you when we do the look in the each of these amino acids here.
Oh, fair enough. That's what I was getting at.
And
Because
so-
amino acids combine to make proteins, correct?
Yeah.
Okay. All right.
So
I get that straight.
this is alanine, and it has a little. This is everything that's the same on it is this amine group and this acid group, and the acids are the COOH. Just a double bond to the oxygen, and this is a single bond to the oxygen here, and a single bond from oxygen to hydrogen. And so, and then over here you have a single bond from nitrogen to these two hydrogens, and that forms the amine group. And so each amino acid works this way. You have, like I was just showing you here, you have the acid part, the amine part, and then this side chain is what actually gives each amino acid its personality. And in order to connect the two amino acids together, it would be to take this OH group here and one of the H's over here on the amine group, and and that creates HOH, H2O.
Water,
Okay,
yeah.
so you remove the water, and it makes that bond. Well, that's a problem, and the problem is in my experiment, I had my amino acids in water, well, water will feed that, so it comes back to disassociate it. And so alanine and glycine, my experiment did nothing.
Okay.
And so I got nothing out of it. Well, there's a couple of other ones I worked with, particularly tryptophan, which is kind of more complicated, and it has this side group that is called an indole. And then there's, I think, tyrosine, and it also had another complicated ring structure over here that hung off of it, and and so when I did the experiment on those two guys, well, it turned brown, and so I said, I got something. Oh, cool. And so what I did is I distilled it all out and isolated the chemicals that were there.
Yep.
And basically what I did is I burnt them. It was pretty much a tar. And
oh, that's your brown sticky goo.
my brown sticky goo is really what I ended up with, and so I put that in a infrared spectrophotometer, and it contained a whole bunch of different things, but it wasn't what I was expecting. And so, I called it my successful failure.
Okay.
Because it was successful because I didn't get what I was expecting, but I learned something out of it.
Oh,
and that's
okay.
what you do in chemistry, is that if you don't get the results you expect, well, then you have to take your theory, discard it, and throw it away and say, well, what is it that really happened here? And so I knew that the sparking it wasn't going to give us any more proteins.
See, that's the thing that always gets me, that even though it's materialistic, why we thought the evolutionary ideas, the stuff you do see contradicts what their predictions are.
Exactly.
And it consistently, but they throw out this particular experiment just like you did, or that particular experiment, but they still hold on to the bottom line scenario, just as they accuse us as creationists, Doug. You basically believe in it, and I said, well, it's not about scientific things, it's about a worldview. It's about saying, yeah, you hold on to a Darwinian book, and I hold on to a biblical book, okay? And you're saying I'm not being scientific, but all your experiments don't work. Things that go against science, Doug. I would say spontaneous generation, disproven. Well, you need that with evolution, okay? Complexity from simplicity, it goes in reverse. And everything that seems to be this chemical thing you're talking about does the same thing. You try to create stuff, and you get brown sticky goo or nothing.
Right.
You don't get anything, yet the very premise that they build all of this on is based on your first experiments. Basically nothing. They're getting something, but they're not abandoning it. And that's what we're accused of, even though I think our what we see in empirical science verifies more the biblical text than this materialistic scenario. So I just wanted to put that back. All these things we're talking about, because it's incredibly complicated. That's why I get to give you the glazed donut look once in a while, Doug. Sorry, I'm going wow. I'm going Bonds. Okay, is it James Bond? I don't know. I'm not so good at this stuff.
Yeah.
I'm looking at it. It's very interesting to you, but I'm going wow. But the complexity of it is what amazes me, Doug, and that all of this stuff fits together and causes things to create shapes that make other things happen. Everything seems to build. It's these little machines or these little organelles or pieces or whatever, they come together. I'm probably using all the wrong terms, but and that's what fascinates me, is that how this all come about by having everything. Oh, that's another one. Everything comes from nothing.
Yeah, we're not even dealing with.
We're not dealing with any of that stuff.
The formation of these atoms at all.
At all. It's like Doug reminds me of the joke. I'll give the joke where the guy says, "I can do anything God does," and he said, "What do you mean? What? I create life. I create life from dirt." And the guy said, and God speaks, says, "Okay, go for it." Now this is, of course, precluded. He's already talking to God, so that the atheist is not really. He's already disabused of his notion. But he goes to get down, and picks up a scoop of dirt, and the voice from heaven says, "Get your own dirt." And the point of it is that God makes everything from nothing. We have a personage, a being, an amazing consciousness that's outside of space and time. Materialism has none of that, and they expect me to believe that everything came from nothing. That's what Big Bang is, by the way. And then from all the simplicity stuff, it gets more and more complex. When everything we see, it goes more and more, it breaks down, it doesn't work at all. So the combination of this stuff, because we didn't even go into the stuff we talked about in other shows, the way life forms, the way the proteins come together, and the way amino acids come together.
Well, I was about to talk about that, Rick.
See, I segues right into it for him. I got right. See, I gave him. You thought I was going to talk, did you? Did you? Oh, I was trying to be good, but now we're back to you, Doug. Go for it.
Well, it's a situation where it is possible for chemists to combine amino acids together to form proteins. And they have a place, they put in their textbook the solid phase peptide synthesis, which is where they go through a series of chemical experiments, just attaching one amino acid at a time to each other through a solid, but it's not in water. It's they've got to take the water out and attach it and make sure it doesn't attach to the wrong end of the amino acid. And so there's a very complicated process that they go through. It takes months of planning for them to do a simple protein like insulin.
So even if they did something that actually created life, are they showing that non-intelligence created life? Is that what, I just thought I'd throw that in there, they're still, but that's the other thing.
You don't believe in intelligent design?
Yeah.
Well, did you, then you believe that your brain evolved. And if your brain evolved, can you know anything about anything?
Correct. Well, that's the other thing, Doug, we talked about it a little bit earlier, but they're taking the water out. Everything we see now, they're looking for planets with water. The most destructive thing there is until you need it, till it's actually fully formed. It can't break. It can't make anything come to fruition in and of itself. It breaks things down. So that's another anti-scientific idea. But that's what they're looking for: water planets. This old life.
Now, I talked about the weak bonds because each amino acid adds meaning to the shape of a protein, and here's arginine, and this is one of them that is called hydrophobic, and it's because it has a side chain on it that has a long chain of oily
so it repels water?
It repels water.
Okay.
And so these guys tend to congregate together if they're near each other in the protein, and it bends the protein in a certain shape. And so and then there's other ones. Let's see if I can find one here. This one, cysteine, has a sulfur in it, and sulfur likes to bind to other sulfurs in it. So what happens is that if there's two sulfurs in the chain of amino acids, and they're some distance from each other, it'll bend around and then connect over here. So it's bound together like that. And it's not easy to get that to actually work unless there's a mechanized way to get that to shape. If you create a protein in the test tube in the laboratory, it's going to fold up any way it wants. If you want to have it actually useful in life, it's got to fold up in a certain way.
Right, okay.
And so that's the disulfide bridges. Let's find another one over here. Let's see what this guy is. Oh, this is another one with a oily chain on the end. It's leucine. And then this one, it's got, and what this one is, it's got this fat molecule on the end, histidine, and it sort of gets in the way of stuff, and so it moves stuff away from it.
Okay.
And so
all of these have purposes that make things happen.
Right. This one is lysine, is another oily chain one. Glycine is just got nothing on it, really. And valine is another oily chain. I keep finding the oily chain.
You do, yeah.
Okay, here's one. This one's got a it's got two acids on. Well, this one is the one that likes this. The ion got an ionic end of it. It's got one that's ionic and one that's not. So they call it a zwitterion. Yeah,
Oh.
and so the positively charged on one end, negatively charged on the other end. So the chemistry of this is really fascinating if you get into it. I'm not going to get into it too much here, except it's going to tell you that when you fold a protein up, it folds up in a certain way that has meaning. And there's millions of ways to get it wrong, okay? You can, and unless there's actually a mechanism within the cell that does this on a microscopic level, and there's these mechanisms that as it's forming, it'll fold it up properly. This is really amazing, and it's taking the DNA molecule, RNA molecules, and there's different types of these, but they all help to form what is called life. And it takes DNA to form proteins, and it takes proteins to form DNA.
So which came first, the chicken or the egg?
And so we have a big problem here, fellows, when you're trying to describe the origin of life.
We didn't talk about the one thing, just a little sidebar, but the amino acids, is it the amino acids? They're always right-handed, or they're always left-handed. Is that what it is?
That's correct.
In life, if you try to make them in a test tube, they can be equal ones, and they kind of mirror each other. But in life, it's always left-handed. It's never right. You never use the right hand. Why is that, Doug? Do they have any idea why that? Why?
It's impossible to separate it chemically. It's a real problem for evolution to explain. In fact, I found a big book in the library one time, a symposium on that very question, where they tried to come to a conclusion, trying to exploit some slight difference between the two different sizes. And
so even before we get to the proteins, the amino acids have insurmountable problems for an evolutionary scenario.
There are barriers at each chemical step. To form these. We're not even talking about forming the more complicated amino acids, which they haven't found a mechanistic way to do it. It's just something that's not really very easy to do. So anyway, I'll show you this complicated one, the tryptophan, which is the one I studied the most. If you want a real chemical name for this, it's called alpha amino beta indole propionic acid.
I'm going with tryptophan.
Tryptophan is the amino acid that makes you sleepy.
Is it tryptophan or tryptophan?
Tryptophan.
Okay, because you have PHAN there.
PHAN, yes.
Okay, okay.
We'll see you next time on Revolution Against Evolution.